Proving discriminatory power of the dissolution method is the topic for this video. Dissolution testing is product specific, and this dissolution testing gives important information and understanding about the in vivo behavior of the formulation. So every batch should be bioequivalent to the clinical batch or bioequivalence batch. That's why the dissolution test is required to be discriminatory. Dissolution of the batch should discriminate between the bioequivalent batch and inequivalent batch. I have made many videos on this topic, and I found the dissolution discrimination a very important aspect of the product development, and that's why I keep on learning and I keep on making the videos, and I assure that this video will help you to understand about the dissolution discriminatory power. It starts with the identification of the critical parameters which are related to the process, material and other formulation variables. So CPP is like, if you consider the tablet formulation, it will be granulation, drying, compression forces, hardness. CM is like particle size of the API, excipient grades, and other variables like binder concentration, disintegrant level. These can be varied to study the impact of this variation onto the dissolution. This impact is required to be studied because it has direct impact onto the dissolution, and dissolution in turn has impact on bioavailability. So overall, the dissolution test gives you important understanding and information about the safety and efficacy of the formulation. It helps to establish robust, reliable dissolution methods. Establishing method discrimination using the real batch variations. So you can use DOE. You can also use one factor at a time approach to modify one variable keeping the other constant and evaluate the dissolution impact to refine the method sensitivity. Batch variability testing. Use real-world batch differences to assess the method robustness. Ensure reliable quality control during routine manufacturing. Understand that the dissolution specifications are based onto the bioequivalence batch. There are some guidelines which provides you the references for deriving to the dissolution specifications, and the bio batch clinical batch dissolution should be considered as a reference for making the discrimination and also for making the dissolution specifications. So we need to create a batch which may not be bioequivalent to the reference formulation, that is bioequivalence batch. It needs a creation of mismanufactured batch or bad batch, which helps to refine the dissolution method limits and identifies impact of variations in the key parameters. So how to create the bad batch? You can change the CMS like particle size of the API. You can change the formulation variables like disintegrant concentration, binder concentration. You can also change some process parameters like hardness, compression force within 10 to 20% of the specification limits. It should not be like that you are directly removing the disintegrant or you are changing the manufacturing process from wet granulation to dry granulation. So always understand that the meaningful changes should be there, and these changes are 10 to 20% of the specification limits. Evaluate the dissolution performance to set the discriminatory thresholds. Setting a discriminatory dissolution specification. For example, I have given here target dissolution threshold. Q is 80 to 85% in 15 minutes. Consider Q as 80%. So in S1 stage, it will be 85% release. Identify suboptimal formulations like compression defects or excipient impact onto this threshold. Then prevent the poorly performing formulation for passing the QC. This is the main purpose, and it ensures correlation between the batches with in vivo performance. Establishing IVIVC and discriminatory dissolution method. So always remember that IVIVC is required to be there between the bioequivalence batch and the routine production batch. because the specifications are derived considering the bioequivalence batch dissolution, and that's why the discriminatory dissolution method should detect the bad batch at the QC release. In vitro in vivo correlation is critical for ensuring the bioequivalence. That's why always have understanding about the IVIVC and discriminatory dissolution method. Use available in vivo data or demonstrated methods discriminatory power. Focus on the critical formulation and process variables to refine the dissolution methods. Utilize in vivo data for IVIVC. If you have in vivo data, then use it to correlate dissolution behavior with pharmacokinetics, and it will help to strengthen bio justification and regulatory acceptance. Also, it will help you to understand the effect of dissolution onto the in vivo performance. And if there is no in vivo data available, then show method differentiation using quality variant batches. Establish meaningful dissolution specifications based on the real batch variations. You can do some variations and you can demonstrate that the method can detect the mismanufactured batches. Biobatch dissolution profile as the foundation. So as I have mentioned, biobatch data, that means bioequivalence batch data, serves as a benchmark for setting the dissolution criteria. Ensure method reflects real world product performance. For example, if your biobatch or bioequivalence batch was showing 90% drug release at 15 minutes, so you have to set the specification for Q as 80% at 15 minutes, and then you should discriminate the batches with this 80% dissolution in 15 minutes. Then how to set Q value formula? So mean dissolution results with 12 units minus 10% at the time point where more than or equal to 85% drug release occurs. So if your biobatch is 90% in 15 minutes, then 90 minus 10%, it will become 80%. Set the Q value as 80%. It guarantees robust clinically relevant dissolution performance. Always understand that you need to have specific Q point specification. If your Q point specifications are too wide, then you will not get proper discriminatory power of the dissolution. In summary, we can understand and say that IVIVC strengthens the regulatory acceptance and bioequivalence prediction. Critical formulation process variables must be assessed systematically. And dissolution discrimination is very important to enhance the quality control. Always use biobatch dissolution profiles as the foundation for setting the specifications, and make the meaningful specification based on the biobatch results so that you can develop a discriminatory dissolution method and you can prove the power of the method. In conclusion, a well-developed dissolution method ensures consistent drug performance, and you can say that every batch which is going in the market will serve the purpose, will be safe and effective as like the bioequivalent or bioequivalence or clinical study batch. Regulatory authorities favor robust science-driven specifications. IVIVC backed dissolution settings or testings optimizes the formulation success. If you can show that though the dissolution is on lower side or higher side of the specification, still the in vivo performance is same, then you can have some relaxed dissolution specification. So based on IVIVC and based on the bioequivalence batch or clinical batch dissolution, you should set the dissolution specification, and then you should prove the discriminatory ability of the method or discriminatory power of the dissolution method for the set specification. So the dissolution specification will be meaningful and will be patient centric and will be clinically relevant. So I hope this video help you to understand about the discriminatory power of the dissolution method and how you can prove it. Thank you for watching the video, and I request you to please do like, share and subscribe to this channel. Thank you.